So welcome everybody. I'm still Jim Curran, Dean of the Rollins School of Public Health. Welcome to the 25th anniversary of the Women's and Children's Center Maternal and Child Health Lecture Series. And I'd like to, before I get the chance to introduce Dean Williams, I'd like to acknowledge the importance of the Women's and Children's Center to the world, as well as to Emory and the Rollins School of Public Health. And Carol Hogue, who is the founder of that center and the director of the center, will be awarded this year at graduation the Thomas Jefferson Award, which is the highest that's the highest honor given to an Emory faculty member by the university. And there's one award each year. And when you look at the list of people at Emory who've received this, they are among the very top, most respected people at the entire university. So congratulations to you, and congratulations to your 25th anniversary. It's my honor to introduce today Michelle Williams, who is the Dean of the Harvard School of Public Health. She and I were just talking. She told me that she would get even with me if I didn't make it short. She's a woman who was trained at Princeton, Tufts, and Harvard, and is back at Harvard after a very successful career as a professor of epidemiology at University of Washington, and is a world leader in studying maternal and perinatal epidemiology. And I think all of you have the, maybe you don't, have the bio of her. But she has hundreds of publications and dozens of grants, even though she's a dean. And she has done tremendous research looking at the in the areas of biomarkers and placental abruption and other types of perinatal outcomes. The one thing I would like to say about her is that a lot of you were at the lecture that David Williams gave, no relation, but only related by also being at the Harvard School of Public Health. And I'd known him for quite a while, and we were sitting in my office, and I said, what do you think of your new dean? And he said, I'm crazy about her. And he said, I was in a room with faculty, and when she was announced as the dean, everybody stood up and applauded loudly and just cheered and cheered and cheered. So she is truly the faculty member's dean, a great research colleague, teaching colleague, and a great colleague as dean of the Harvard School of Public Health.
So I have no reason to get back at Jim because you kept it short, so thank you. And thank you all for coming. I want to say this feels like a bit of a homecoming because in 1987, I was a graduate student at the Harvard School of Public Health, and in '86, I met somebody who was teaching at the summer epi program that Ken Rothman ran on the Tufts campus. And I met the first reproductive epidemiologist I'd ever met in my life, and that was Lowell Seaver, who's sitting right here. And I remember, along with another classmate, we kind of literally sat at his heels in the grass outside on the Tufts Medford campus, trying to learn from him everything we could about what exactly did a reproductive epidemiologist do. And we learned from him that it was what we wanted to do. It's what I wanted to do. And once I figured that out, I think the next day I asked you, well, how do I get to follow you back to CDC so I can actually see you in your ecosystem? And Lowell Seaver generously invited me to come and spend the summer of 1987 in the division of birth defects headed by this gentleman over here, Godfrey Oakley. And that was the beginning for me of a journey where I found my purpose in life. I had found the discipline I wanted to embrace and work on, but I had not found the content area that I wanted to commit myself to, although I knew as a person who studied developmental genetics, it had to be developmental in scope. And really, sitting right here is the person who helped open that door for me, and thank you. I didn't know that I'd have a chance to do that today. And thank you, because your leadership in creating a division that allowed people to bring in students with, at the time I had nothing to offer really, was a great thing. So thank you. And while I was hanging out in your division, I did hang out a little bit in the Division of Reproductive Health. And while I wasn't a card-carrying member of that group, I do remember spending lunchtime in your division, going to seminars hosted by your division, and even the second summer when you were hosting big meetings to talk about HIV/AIDS and what it meant as a public health problem in urban communities. I remember piggybacking on and getting a pass so I could be a part of that. And so, you know, I'm talking to a group of people who, these three individuals played such a role in my early part of my career, that you all are so fortunate to have them here because their reach in educating and preparing next generations of leaders is far beyond the physical space that they occupy. So thank you all for what you've done. So I get a chance to talk about some work that I've been doing over the last, let's say, 2 decades. It's been an obsession of mine to understand the epidemiology of placental abruption. And I get to tell you a little bit of a story about how I've continued to try to unpack this mystery using the tools and principles of epidemiology. Let me tell you my bias. And my bias is I believe that when medical complications of pregnancy occur, that they're not isolated events. I think that they occur as a result of failures in maternal metabolic and vascular systems. And that is the underlying premise and bias that I take when I approach the questions of why. Why does it happen? And it could be placental abruption or preeclampsia, hypertensive disorders of pregnancy, or it could be gestational diabetes, a disorder that looks a lot like type 2 diabetes, but it happens in pregnancy, and after the pregnancy, the REC symptoms go away. But interestingly enough, women who have gestational diabetes have a very high risk for developing type 2 diabetes later in life, and not a lot of us have spent enough time trying to understand that transition because there's an opportunity for prevention that we just keep missing. And then, of course, preterm birth. And I also like working in this space because this is the one place where you've got 3 unique genetic systems cohabiting: the maternal genome, the fetal genome, and the placenta, which might not necessarily always represent the intersection of mother and father. So when I talk about placental abruption, and it's a general audience, so I want to spend a little time describing this thing that I've been obsessed with for more than 20 years. So I wanted to share, I'm going to move, okay? I want to share a bit of the perspective because usually epidemiologists start with the definition, and I've been talking a lot about my transition in trying to be more communicative to broader populations. And I think better to not start with the disease defined by the experts, but the disease defined by the patient. So I'm going to share you a couple of perspectives. And this is from a woman who developed placental abruption at 24 weeks gestation. And she's describing, he was so tiny, I had only seen pictures which never captured his size. Jenny, the nurse, explained everything that was hooked up to him. It was so much. Then the doctor talked to us. Julian's blood pressure was low and the meds were maxed out. His infection, rare for his age, was bad. Surgery could fix it, but it would mean losing most of his bowel. His brain bleed had been so severe, it had moved his brain. He was not breathing at all on his own. That's the patient's appreciation of the condition she developed, 24 weeks gestation. And imagine doing an interview and asking, did you have placental abruption? Yes, no. I don't think she'd answer that. But she did, and this is how she perceived the condition. Another one, and I won't read this, but just note, you know, she's 39 weeks, so her event happened at 39 weeks gestation. She has an 11-year-old, a 9-year-old, and a 1-year-old that she's cuddling with. And she's having a nap, and she woke up with pain, which is one of the early symptoms of this condition. And the pain's immense, and she had just been for a checkup the day before. A sudden event, a mother with 3 children, And she just had a checkup and everything was fine. So abruption, the word as we can find it in the literature appears in 1926, and it's abruptio placenta from the Latin, which means, which is basically breaking away from, and it's the placenta breaking away from the decidua. Trying to study it as an epidemiologist has always been challenging, and in fact, clinicians have always dissuaded people from studying it because they can't come to a consensus on describing it. And this is one of those places where we end up undoing a lot of goodwill in trying to understand complex, heterogeneous, but important conditions. So it is the premature separation of the placenta from the uterus before the delivery of the fetus. It can and often is accompanied by hemorrhage at the decidual placental interface because of that abruption, that breaking apart. And it complicates roughly 1% of pregnancy from the best that we can tell of all pregnancies. It's ostensibly a clinical diagnosis, and the research definition has been constructed in ways by people who've been tenacious and have persisted on trying to say this is important. We should try to come up with a standard that helps us understand how to identify and then study the condition. And essentially it's been characterized by the vaginal bleeding in the second half of pregnancy with non-reassuring fetal status, uterine tenderness, that pain in the tummy that women talk about, and then on evaluation, evidence of adherent blood clots or retroplacental bleeding. And technology doesn't always help with the diagnosis, but carefully done sonograph can identify symptoms and signs of the condition. Looking at the data that are available, you can see the occurrence of abruption varies across these different sites. The best global estimate is about 1% of singleton births. The timing, I want to talk a little bit about the timing because I find these graphs to be quite interesting. When you look at the patient perspective, you had some patients who developed abruption early in the second trimester, others quite late. These are three different populations from the US, from Finland, and I forget where this one is from, but you can see the attack rates at different gestational age showing quite similar patterns where the peak is in the second trimester and then attenuates down as pregnancy goes on. I want to talk a little bit just to describe the pathophysiology briefly. It looks, when you study the pathophysiology, that there are chronic and acute processes that lead to abruption, and the evidence listed here for some of the chronic processes, you can see very early evidence of impaired placentation by ultrasound or sonograms being done so that you identify either impaired blood flow, uterine blood flow, or actual cheesy-like lesions in the placenta that precede abruptions months before they actually occur or weeks before they occur. Other evidence of the chronic process would be evidence by biomarkers of oxidative stress, looking at placental proteins and looking at platelet aggregation and endothelial cell dysfunction. On the acute processes involve thrombosis or shear stress. So when the water bag breaks, the amnion, chorion that contains the amniotic fluid and the fetus breaks, that causes a sudden decompression, allowing for a shearing of the placenta from the decidua. And then inflammation, evidence of cytokines, acute inflammatory response, or even acute vasoconstriction as a result of cocaine-induced vasoconstriction, or transient systemic nervous system activation like rage. And I'll come back to rage for a minute because it's what motivated my thinking of using the case crossover method to study triggers of abruption, acute triggers of abruption. Why it's important? It's important because the maternal complications are not inconsequential. Hemorrhagic shock, pulmonary edema, coagulopathies, renal failure, and death. And in some countries where tertiary care is not always easily accessible, continues to be a very important cause of maternal mortality. On the fetal infant side, it's a very important contributor to preterm birth. It's the outcome more people like to study, but I think this group knows that that's a very heterogeneous group of preterm births, and to really appreciate and understand the pathways and mechanisms that lead to preterm birth to enable us for prevention requires us to look at some of these important contributors, and abruption, I argue, is one of them. Growth restriction, stillbirth, and a variety of developmental disabilities that are associated. So the other thing, and I shared with you my bias, I believe that what happens in pregnancy tells us a lot about what happens to women as they age for non-communicable diseases of aging. And the data are emerging that abruption is indeed an important harbinger of maternal cardiometabolic disorders. And I put here on the slide a number of studies that have emerged that have followed women with a history of abruption, or have looked back, looking at records using registries to identify associations that would suggest a history of placental abruption in pregnancies associated with fourfold increased risk of cardiovascular mortality when you compare them with women who did not have pregnancies characterized by the complication. And in another study, all-cause mortality twofold higher after a 15-year follow-up when you looked at women with the history and those without. And even just looking at cardiometabolic risk profiles, more evidence of dyslipidemia, hyperglycemia, and persistent inflammation, often measured looking at C-reactive protein 6 to 9 months after the pregnancy. Another patient perspective I just wanted to share. Suffered a complete abruption, delivered by C-section. Again, the focus on the baby's health, and this was a term abruption, or happened close to term. And the mother very concerned about the developmental trajectory of the child, and is reporting here as that having survived the complication in the perinatal period, her son walks, talks, laughs, and is meeting the developmental milestones. So women who survive this carry the burden of worrying about the developmental trajectory of their surviving kids. Some of you who are golf fans might recognize this person. I think it's always important if we can have a champion help promote our cause, especially in reproductive health. And this is Annika Sorenstam, a retired golfer who had placental abruption at 27 weeks. So she was one of the early ones. And this occurred in 2011. She was very public in sharing about her medical history. And this is her celebrating her son's first birthday, happy, healthy 1-year-old, relieved that her son, delivered at 27 weeks, is thriving, healthy, and reaching the milestones. So that's abruption. And just quickly, when I started looking at this as a doctoral student in 1986, 87, thanks to Lowell Seaver, bringing me into the space of reproductive health, and I wanted to find my niche, I ended up studying the delivery interview program database. That was a large March of Dimes funded study at the Brigham and Women's Hospital, 13,000 pregnancies. And it was because it was so large and there was 100 or so abruptions that I had a chance to start to dig away at what the epidemiology was with that database, but also looking at the literature. And when you look at what the risk factors are, I don't know about you, but if you're studying cardiovascular disease epi, a lot of these overlap and overlay, and that just fed into my bias that when we're talking about medical complications of pregnancy, we don't need to divorce ourselves from those people who are studying chronic metabolic disorders in non-pregnant individuals. And that just kept feeding my bias that that's how we should be thinking about this. One prominent risk factor that was very strongly associated with abruption in every study, essentially, was chronic hypertension, that people with pre-existing vascular disease, chronic hypertension, have a very moderate to high risk of abruption, and that just kept feeding the bias. This is another way of representing the data available in 2006. This was published in 2006, and you can see the risk factors look a lot like risk factors we would see elsewhere. Because we're talking about pregnancy, there would be some that's specific to pregnancy. But smoking, chronic hypertension, vascular drugs or substances that immediately impact the vasculature. So I thought, let's take a look at some other risk factors that we don't typically look at in perinatal epi in relation to placental abruption. And over the years, I'd had students and fellows just take a look using data available at risk factors like, well, migraine is an important risk factor for stroke and cardiovascular disease in non-pregnant populations. So we asked, what about migraine and placental abruption? And these are mostly case control studies because not the best design for causal inference, but for a disease that people don't always think about. We can't get a coalition to get together to do a prospective, efficient collection. You do what you can, and these are mostly case control studies, and you ask people about their lifetime history of migraine, and you try to do everything to minimize a bias, to improve inference, and these are studies that show when you look and ask the question about migraine history, women with abruption compared to those without have an elevated risk. And even if you're asking about tension headache as opposed to migraines, you can see elevations in risks. I've always been concerned about women entering pregnancy either with a history of mood, psychiatric disorders or anxiety disorders and their pregnancy outcomes. And here, this was an undergraduate student who contributed to looking at this in a cohort of, in a case control study of Peruvian women with a history of mild depressive symptoms, moderate depressive and severe depressive symptoms in early pregnancy, having increased risks for placental abruption in a cross-sectional conducted study. We've looked at sleep disturbance because circadian misalignment and sleep disruption, acute and chronic, have important roles in abnormal metabolic response and cardiometabolic diseases, and we've started to look at what early pregnancy complaints of short sleep or sleep insufficiency looks like in relation to later risk of abruption. And these were early preliminary cross-sectional studies that say there might be something here that we should continue to interrogate with longitudinal data. Vital exhaustion and violence, intimate partner violence. I was particularly interested in not just the psychosocial stress of violence and its role on maternal placental function, but also immediate trauma from blows to the abdomen. And those, unfortunately, are not uncommon events. Violence happens in pregnancy, and some abruptions end up in the emergency room as a result of trauma that is intentionally meted on women. The genetics of placental abruption, quite interesting, very difficult to study because you need large samples. And what's come early on from the studies that have looked at abruption, and these are generally small case control studies that were in 2008 summarized in a meta-analysis published in Epidemiology. At that time there were 22 articles, and from the 22 articles the polymorphisms that were most commonly identified with the condition are in the vitamin B, folate, B vitamin, folate, metabolism, and endothelial function pathways. And for a long time, people kept working on looking at genes in this pathway as important genetic determinants of abruption. But what's really needed, and we know this now, is to organize ourselves in ways to allow for a more agnostic approach to identifying the genetic determinants of this disease. The way we are aligning, other sub-content area experts are aligning themselves to study the genetic architecture of severe mental illness. It requires large teams coming together, and not hundreds of samples, but hundreds of thousands of samples to really capture the human experience, and then have the sample size and power to look at the genetic architecture. This on the slide is a representation of our beginning efforts to do this. It's kind of a field of dreams approach. If we build it and we show where we would like to be when we have the hundreds of thousands of abruptions, and we show the framework of how we would interrogate this very large collection of data sets by doing the type of agnostic genome-wide association studies, and look for those markers, those genes and pathways that might inform inform causal thinking, but also inform where therapeutic and interventions may come. And this framework exists in other areas of our work, most prominently the work that's being advanced in the area of psychiatric epidemiology, where very large samples are being brought together, and people can start to look to see if there are real heterogeneity and differences in the genetic architecture of schizophrenia versus autism. And where is there commonality and where are there differences? And it's a bit heretical to think about combining autism and schizophrenia, but for these kinds of genetic studies where we're really trying to identify what's common and where the overlaps don't exist, you know, I am a proponent of putting the information we have, putting the genomic data together and interrogating in an agnostic way. I also believe that it doesn't have to be a false dichotomy, that one can also take prior targets and interrogate them very carefully and rigorously and robustly using candidate approaches, and not maybe candidate genes, but candidate pathways. Another thing I wanted to share with you that motivated our thinking, strokes and MIs happen following a circadian pattern. And I used to walk around naively saying, well, abruption's kind of like an MI, but it happens below the waist. Right? It's a vascular accident. It just doesn't happen in the brain or in the myocardium. It happens in the placenta. So I was curious about the circadian rhythm. Can we find a circadian rhythm in some of these acute pregnancy outcomes the way our friends in cardiology or in neurosciences find them for stroke and MI? And I'll tell you honestly, the data are just not quite there, and there's some unique challenges. And the unique challenges for us is abruption will likely be the pain and the bleeding that brings the woman to the hospital. So playfully we said, well, can we figure out from asking the women about the onset of the vaginal bleeding and the back pain that promoted their coming to the hospital at the early possible convenience. We know it's not perfect data. We know it's, you know, it's what they can tell us. And we basically plotted out, you know, when they reported the onset of the pain. And A panel here are women who experienced abruption at term, and these are the ones who experienced abruption at preterm. And you see more of a circadian pattern that looks like the cortisol release cycle in the term abruption, but there seems to be a circadian disruption or some side of circadian rhythm in rupture of membrane and placental abruption. And this is a placental abruption for the preterms. We wondered about whether or not there might be genetic markers that we could look at, and we had an opportunity with data from Peru to look at 119 SNPs in 13 candidate genes that are known to control circadian rhythm. And we did a candidate gene approach of clock-related genes, created a genetic risk score where we essentially combined the effects of multiple SNPs and looked at their relationship using a weighted genetic risk score on the risk of placental abruption breaking the scores down by quartiles. And the first quartile, the lowest score, upper quartile, highest score, you see a gradient, a risk going from 1.8 to quartile 2, 2.8, quartile 3, and a 5-fold increased risk. And this is just a plot of the score as a continuous variable, which is the log odds of placental abruption in relation to the weighted genetic risk score. So, you know, in our playful use of the available data, recognizing the limitations, we come to think about abruption. Although it's very, very rare, we recognize it's a devastating placental complication, has adverse maternal and perinatal outcomes that are quite considerable to the patient, to her child, and I'd submit to society because of its contributions to preterm birth. Traditional risk factors, as well as the novel ones that we've started to look at, point to a complex disorder with both acute and pathophysiological processes, chronic processes, and the parallels in the pathophysiology and the epidemiology between humans. And I'm not, because of time, talking with you about abruptions in non-human primates, but the data of abruptions in non-human primates exist, and it's really interesting and a lot of fun. And one of my pet goals would be that I could team up with someone who's obsessed with abruption in non-human primates, and we could work on sort of a loop where we generate hypotheses and test in humans and then test them mechanistically in the non-human primate model. But the epi of abruption in non-human primates, it's a small body of epi, but very reminiscent of what we see in humans. And then it's still a relatively underdeveloped area, and their interesting resemblance to CVD, cardiovascular disease, and acute MI. So that got me thinking about how I can use a study design that is very rarely used in perinatal epi, which is the case crossover study. And the case crossover study is a type of study, if I can briefly just simplify, it allows you to not ask the why me that most of our study designs ask and answer. The case crossover allows you to say why now? Oh, you know, why did it happen now? What was I doing that triggered this event? The crossover design is, again, a scientific method to allow the answer to the question, what is the event that, abruption, that was triggered by an unusual activity before the abruption? And this, and so it's really why now? Why did that event happen now, as opposed to why me? What are my risk factors or predisposition, genetic or otherwise? So this is a design I sort of watched get put together by faculty in my department when I was a student, by Malcolm McClure, who is now in Canada, in Vancouver. And it's a general approach to compare the exposure immediately prior to the case-defining event with the individual's exposure at a referent period. So it's been used extensively, along with Murray Middleman, who's a faculty member at Harvard and was a former classmate working with Malcolm on this, and has been used in many different settings of acute cardiovascular disease in the last 2 decades. It's a self-matching design. In the analysis, each person serves as their own control, and the concern for confounding is minimized because of that self-matching that happens for time invariant characteristics. I just want to show you on this slide that it's been used to study these kinds of exposures here: heavy acute physical exertion, alcohol binge drinking, sexual activity, anger, rage, or bereavement, and extreme temperatures. And these are the ranges of outcomes that have typically been looked at. MI, injury, preterm labor. Sonia Hernandez-Diaz has done a nice study, small pilot study on this, and I wanted to do it on abruption because I think it's the kind of condition in pregnancy that is prime for using this kind of study design. Just want to walk you through a very nice paper. If you haven't thought much about this design or looked at it, this paper I find by Anna Balin is beautifully written, very clear, on an example that looks at triggers of non-fatal MI in Costa Ricans in relation to acute exposure to physical exertion, sexual activity, and infection. And for this, I'm just going to show you the physical exertion example. So it's a study. She is enrolling and interviewing individuals who survived an MI so that she can ask them questions about what were they doing hours before the MI symptoms, and then analyze the data in ways to help compare the activity at the triggering activity, the participation in the activity that triggered the event in relation to what a referent period where that activity may or may not have been occurring. And what she found is, just focusing on the physical exertion, that the triggering effect of heavy exertion was the relative risk for MI in the hour after physical exertion was almost 5-fold. Okay? And that's looking at all 53 cases reporting the timing of physical exertion in a reference period and in the window of risk. She then stratified the information and said, okay, of the 53 people, how many have been relatively physically fit or unfit? And she assessed fitness based on their routine energy expenditure in vigorous activities. Okay, so it's their baseline fitness now, and given your baseline fitness, what is the triggering effect of the heavy exertion on your risk for MI? And the most sedentary people are the people who are less likely to be expending routinely energy on vigorous activities, this group of 28 here. They're the ones, when they engage in acute physical exertion, their risk for MI an hour after that exertion is 27-fold. Now, these are wide confidence intervals, but what she begins to do here is say acute physical exertion across the board is associated as a triggering effect for MI. But that triggering effect is even more profound for people who are least fit, fit defined based on routine energy expenditure. And I thought this is really interesting and cool, and it allows us to begin to refine the public health message. So I wanted to see if we could do something like this in placental abruption. And it took me 5 years, but I did get funding to look at triggers of abruption. And study section said, well, if you're going to look at triggers, do a traditional case control study as well and look at the genetics. And I said, yes, great. I'll just double the budget. And this was back in the old days, and it actually did get funded. So we designed a study where the cases themselves are going to stand alone in the case crossover design. And if we were not going to do a traditional case control study, all we would do is just get the cases alone. But given the endorsement of this great study section, we also were able to make the case for doing the traditional case control study. So we got maternal as well as fetal DNA buccal swabs to allow us to do both the genetic as well as the trigger studies. And we chose to do this work in Lima, Peru, because I had been working there for 17 years, and they're large hospitals, and we could put 7 together in a coalition and have a number of 30,000 deliveries a year. And when you're looking at something that occurs at barely 1%, big numbers and friends in big hospitals are very important. And there are not a lot of Grady memorials around. I learned that. So we put together these 7 hospitals, and we've been working with these 7 hospitals and the Ministry of Health in Peru for 20 years now, and it's just been a great place to get these kinds of studies done. So our goal was to look at triggers, but also to try to begin to continue to build a framework where we can do the very important work on looking at the genetics of abruption. Just the setting, it's been a privilege working in Peru with partners in the Ministry of Health and clinicians and members of medical societies. We started data collection in 2013, and it's ongoing. We're identifying cases through this massive network of health providers who are making the information available for us to do the surveillance of intake information so that we get as close to the diagnosis as we can. And I'll skip that in the interest of time, but just try to tell you, we did a data freeze in the first 440 cases, and I just want to show that these are not trivially impacted patients. The birth weights of the cases are quite low, 1,800 grams on average. C-section rates very high. 63% of these abruption cases are preterm, and 64% are low birth weight. And this really is a profile that you'd expect to see for a case group of abruptions. And it's a generally low-income population. Just quickly, this is an interim analysis of our trying to get at the triggers piece. And I show you here what women report about physical exertion and abruption. And our hazard period for this study is 6 hours before the abruption, 6 hours before the bleeding or the pain that brought the woman to the hospital, this strict definition. And just to orient you, this is the relative risk scale on log scale, and heavy exertion and moderate exertion are the two exposure that I'm sharing with you here. And for heavy exertion, we defined this as that they were running or pushing something heavy or pulling with all their might and grasping at a level where they were also exerting themselves to the level of sweating. And in doing our best to try to characterize this, the women who endorse engaged in heavy exertion in a 6-hour window before abruption, their abruption risk is 2.8. And those endorsing moderate exertion, such as normal walking or house cleaning, like cleaning windows that result in their breathing deeply but not gasping, was 1.9. So these are preliminary data, wide confidence intervals you can see, but we're trying to do what our colleagues in cardiovascular epi did in looking at, when you look at physical exertion as a trigger, is there any signal? And what we need to do with the larger data set now is evaluate the extent to which these relationships are similar or dissimilar for women who are habitually active versus those who are habitually sedentary. And you can see how we would begin to frame a discussion that many pregnant women have around routine physical activity in pregnancy. And for far too long, American Medical College of Obstetrician and Gynecologists have been silent on messaging to women what they should do about their routine physical activity levels. And we're only getting better about that in the last few years. This one is emotion, and I'll just quickly, this was basically asking women, and I didn't indicate how we operationalized panicked, but we asked about panicked, and we asked about feeling very anxious. And the women who were reporting that they were feeling panicked 6 hours before the event, and this is in the control period, which is the last week of the pregnancy. Those reporting panicky feelings 6 hours, abruption risk 7-fold. Those feeling very anxious 5.8. Difficult to interpret at this stage because are they panicking, feeling anxious because of symptoms they were experiencing? Hard to tease out in a study design like this, and honestly I'm not sure how we're going to go forward with trying to report this in the published literature. But what we're trying to get at is the evidence that we see in the cardiovascular world where bereavement really has been documented is as a trigger for cardiovascular events. So our early triggering studies are yielding findings that are not dissimilar from the early case control studies that the early case crossover studies that the epidemiologic literature in CVD epi brought us. I think it's important in this population of pregnancy to think about physical and psychological stressors as potential triggers of vascular events important in pregnancy, and abruption is one. Our estimates so far are imprecise, but we're going to get there. We have close to 900 abruption cases now. If we can look at the triggering event across a context of baseline, like what Anna Baling did in looking at sedentary people and non-sedentary people and whether the triggering yielded the same spike in risks, that will help add some precision to our discussion around risk stratification. And then one of the big criticisms around case control studies, case crossover studies, that you might identify some very large point estimates, like the 4.0 in Anna's study for heavy exertion and MI, but the absolute risk is quite low. And I think interpretation has to be taken in mind that we need to assess the absolute impact of these triggers. Just briefly, I wanted to just share, in thinking about the other things that we try to do in looking at placental abruption, is to integrate new methodologies. And I've been a strong proponent encouraging junior faculty who work with me to bring new tools into the space of perinatal epidemiology. And a young scholar who's working with me managed to leverage some resources that I started saving back in 1993. When I moved to Seattle, Lowell, and I set up shop at Swedish Medical Center, I found out that they were throwing away the serum samples that were collected to look for neural tube defects screening and for chromosomal abnormality screening. And at the time, no one wanted to invest in biorepositories. They weren't calling them that. And no one wanted to invest in cohort studies of pregnancy because they were too expensive. Although they did invest in cohort studies of cancer and cardiovascular disease, you only had to follow these people for 9 months. And really in 91, 92, there were just so few that I said, well, how about I make a deal and I collect the biohazard of the prenatal diagnosis lab. And I talked the perinatology and the clinical chemistry lab to let me take the samples that they were storing 3 months after the patient finished their AFP screening. And they did, and I asked them, can you keep them in your freezers for me, and then I'll get them later? And they didn't know later was going to be like 15 years later. So they started to tell me, you got to move the samples out or we're going to dump them. And I won the epidemiology prize from Ken Rothman for writing a paper on fish oil and preeclampsia. And it came with a $3,000 check. And I used the $3,000 check to buy used -70-degree freezers. So when my samples got evicted, I put those samples in borrowed space in the used freezers. And what I'm sharing with you now is how we use metabolomic techniques that weren't developed or commercialized until recently with samples that I have been storing since 1993. And I didn't get to do the work because I had to do administrative stuff, but at least I got someone to use those stored samples that got me evicted. But then Ken Rothman's prize gave me used freezers, which caused me heartburn because I walked around with a pager on my belt for 10 years. Because if the freezer alarm died at 2 in the morning, I needed to get up out of bed and go down to First Hill and deal with it. And I did that for a long time. But it was worth it, and I'll show you why. So metabolomics, I like the concept. I'm always trying to bring things in. And for me, I see this technique as a way to help us phenotype in ways that are not prejudiced by what the clinician says or what the patient says, but by what the molecules in the bloodstream of the person might tell us about the disease if you get the blood at the same time of the diagnosis, or what the harbingers are, where the disturbances are in the metabolism before the diagnosis. And so I watched people using it, beginning to use it in the area of preterm birth, in fetal growth, in preeclampsia, and in gestational diabetes. And really the use of metabolomics in type 2 diabetes has been really fun and interesting. And I've watched it, and we've deployed it in gestational diabetes. But I wanted to do it in abruption, and we managed to do a small pilot study I'll just share with you. The goal is use some of those 16-week samples and see if molecules, metabolites in the blood, are different from those women who went on to develop abruption versus those who didn't. And these are the people, women coming in to OBGYN visits, getting their bloods drawn for prenatal diagnosis, saving the blood, not optimally stored. It was -20 and -70 for decades. And we sampled for this pilot study 50 women who went on to develop abruption, 51, and 51 who remained abruption-free. And this was done with a reference lab at RTI in Raleigh-Durham. And we did a targeted metabolomics and looked at these molecules that have been well characterized and looked at in other conditions. And I won't go through the analytic approach, but we found a series of metabolites that were associated with abruption and are starting to really try to understand what they mean in terms of their biological pathways. And then also to look at how these metabolites, when you couple them with clinical risk factors for the disease, like early pregnancy, vaginal bleeding, might help us come up with risk prediction as well as trying to understand pathophysiological pathways. Bezu Galai, who's the primary author of this, worked with a chemist, and they evaluated a ratio of 2 acrylcartanines. These are compounds that are very involved in mitochondrial function. And mitochondria, you might remember, is the cellular organelle that is described as the engine, the energy engine of cells. And they basically created a ratio where an excess of one, a deficit of another, is quite predictive of abruption. And when you put the two together, the predictive probability of abruption looks a lot better than when you look at one alone. This is the ROC curve, and it's far from ready for prime time in terms of risk prediction. But it's just a description of where we could go if we can start to understand the metabolites and get better measures of behavior as well as clinical characteristics to help do some risk prediction for abruption. And I think I'll stop there. I just wanted to share the journey of how I've tried to integrate the beautiful methods of epidemiology, the techniques that arrive decades later, and the need to sort of sometimes think outside of the box, especially if you're obsessed with a condition that you feel people don't pay a lot of attention to. You try to draw them in by telling a narrative around what is biologically, pathophysiologically similar or dissimilar, and engage them. And I tell you, the cardiologists love when I say it's an MI below the waist. And I will tell you that I felt free in talking that way because I saw Godfrey Oakley do that when he tried to mobilize people to appreciate what you need to do, what you can do to prevent neural tube defects. Sometimes you do have to practice a little hyperbole. So thank you.